A pleasant study time to everybody. I hope everyone's doing great despite all the pressures for the upcoming final examinations. So, once again, I am Sir Roy, and I will be presenting you with a very brief overview and with the general concepts about your major histocompatibility complex, the HLA or the human leukocyte antigen. So I believe you may have probably heard and have tackled the MHC in your immunology. So when we say MHC or major histocompatibility complex, this is basically a cluster or a collection of genes that are located at the short arm of the human chromosome 6 on band 21. So in general concept class, your MHC determines whether an organ will be rejected or tolerated when transplanted into an individual or to a patient. So in humans, the products of the MHC are called your human leukocyte antigens or the HLA. We have three different antigen classes for the HLA. We have class 1, class 2, and class 3. So as for class 1, human leukocyte antigens, this includes the products of the HLA-A, HLA-B, and HLA-C loci. And the antigens are found on all nucleated cells of the body and as well as in your platelets. So class 1 antigens are involved in the recognition, also termed as your MHC restriction of your cytotoxic T cells, which bear the receptors CD8 for the HLA antigens, and they are associated with the expression of antigen on virus-infected cell or transformed cell. So as for class 2 antigens, this includes the products of the HLA-D, HLA-DP, HLA-DQ, and HLA-DR loci. And they are found primarily on B lymphocytes, activated T lymphocytes, skin macrophages or your Langerhans cells, epidermal cells, and as well as sperm cells. However, also take note that class 2 antigens are absent in resting or naive T cells, in platelets or your on your granulocytes, and what else? Your fibroblasts. So class 2 antigens basically or generally allow the interaction between antigen-presenting cells or APC and T cells via CD4. And this makes class 2 antigens class highly associated with the presentation of antigenic determinants on the antigen-presenting cells. And lastly, for your class 3 human leukocyte antigens, are actually involved with the complement cascade, specifically in complement activation, opsonization, and the generation of your chemotactic factors. And the complement factors involved are your C2, C4A, and C4B, and also your BF, or also known as properdine factor B. Okay, the immunologic role of your HLA falls as the antigens are recognized by the appropriate T cell receptor only in the context of the histocompatibility antigens. So, when we say endogenous antigens, like for example, virally transformed proteins would be expressed on the surface of any cell in the context of class 1 HLA molecule and be recognized by a CD8 cytotoxic T cell. And when we say exogenous antigens, like for example bacteria, would undergo processing by the antigen presenting cell and therefore would be expressed on the surface of the APC or antigen presenting cell in the context of an HLA class 2 molecule and be recognized by CD4 plus T helper cells. So as for the transplantation roles naman, since the HLA gene products are cell surface proteins and are very antigenic, like what you have on your RBCs and those antigenic determinants. It is precisely and really imperative that tissues must be tight before any transplantation procedures or transfusion procedures. So these are some of the disease associations of the HLA. It says here that certain diseases have a propensity for persons with a particular HLA antigen, or it means that an individual having the particular HLA may have the tendency to develop the risk of having these specific diseases. So for B27, there is a propensity or tendency for ankylosing spondylitis and Reiter syndrome, dermatitis herpetiformis for DR3, also Sjogren's syndrome, as well as insulin dependent diabetes, which is alongside with HLA-DR4. C6, psoriasis vulgaris, DR2, good pasture syndrome, and B54, Behçet's disease. So here class is a nice illustration of the basic and a simplified version of the structures of the HLA class 1 and class 2 uh, antigens. So HLA class 1 are made up of two polypeptide chains, a heavy chain and a light chain. The heavy chain is basically encoded by the HLA genes and carries serologically detectable determinants such as the alpha 1, alpha 2, and alpha 3, while the light chain is encoded by the chromosome and carries beta 2 microglobulin. For HLA class 2, the antigens are also made up of a light chain and a heavy chain. The demonstrable determinants here include alpha 1, alpha 2, and as well as B1 and B2 products. Your human leukocyte antigen system is polymorphic, and this polymorphism is actually due to the multiplicity of alleles that are basically encoded by each of different loci. So for HLA-A, it has 23 antigens, HLA-B has 49, 8 for HLA-C, 19 for HLA-D, HLA-DP has 6 antigens, HLA-DQ has 3 antigens, and HLA-DR has 16 antigens. And take note that the chromosomal region encoding HLA-B, HLA-A, B, C, D, and HLA-DR are inherited as N-block haplotypes. HLA typing technique, the lymphocytotoxicity technique. So as per genomic ruling, the HLA typing is performed using the lymphocytotoxicity technique. And nowadays class, and actually lymphocytes are always used as the target for the HLA typing. So in this technique class, we have two modifications. However, we are no longer diving into deeper scale of explanation since this topic is generally not much of academic importance. So for identifying HLA-A, HLA-B, HLA-C class 1 series antigens, a lymphocyte suspension consisting of T and B lymphocytes, approximately 70% T cells and 15% B cells. So while in case of HLA-DR, HLA-DQ, and an enriched B cell, which usually comprises of 70% population, is necessary. And this is because class 2 antigens are not found in resting T cells. And so as for the principles of this technique, the HLA anti-sera will allow to bind to their specific cell membrane target antigens at the surface of the lymphocytes and then preceded by the complement activation by the antigen antibody complex on the cell, which leads to cell lysis. The lysis class is detected by the entry of the dye into the dead cells but not into the living cells, and this is the positive reaction. When no antigen or there is an absence of antigen antibody binding in the procedure, the cells are not stained by the dye, which results to a negative reaction. And in phase contrast microscopy, viable cells or the living cells will appear as bright and refractile, whereas dead cells are or will appear as dark and rather larger in size. We also have here two more procedures involved in HLA, the lymphocyte isolation and the T and B lymphocyte separation. So as for lymphocyte isolation, this is done by using a phycohypake or phycoisopake with a density gradient solution of 1.077 grams per milliliter. And this cell preparation is the one we utilize for the HLA-B-C typing. And then next procedure is the HLA or lymphocyte separation. So in this procedure, nylon wool can be used to separate T and B lymphocytes from one another. The take note class that your B lymphocytes are adherent, and this is due or because of their cell surface properties, and that's exactly why these B lymphocytes stick to the nylon, whereas T cells can be easily washed off from the nylon. And okay, class, actually this preparation or the preparation method, your lymphocyte separation method allows B cell enrichment to up to 70 to 80% in the samples. So to reduce the rejection of transplanted tissue, immunosuppressive drugs are actually administered, such as corticosteroids, cyclosporine, azathioprine, and also radiation. However, all these measures enhances the recipient's susceptibility to opportunistic infections or neoplasms. There are at least three types of rejection reactions that can take place following a transplantation procedure. Number one is hyperacute rejection, and it occurs within hours or days following the organ transplant. And this is due to the attack of preformed antibodies to the organ. So this category of rejection class, there is a rapid intravascular spasm and vascular occlusion, and the organ is not perfused by the recipient's blood. So number two is acute or accelerated rejection, and it occurs within 10 to 30 days after the transplant. And unlike your hyperacute rejection, an accelerated rejection is mainly due to sensitized T lymphocytes. So when we say T lymphocytes, when T lymphocytes are involved, it means that this is a cell-mediated immune response. So there is infiltration into the graft, especially around small blood vessels of small lymphocytes and mononuclear cells, along with some granulocytes, which causes the destruction of the graft. And that is according to the Essentials of Medicine by Cecil, fourth edition. And lastly, number three, we have chronic rejection. And unlike the first two, a chronic rejection takes a longer time frame of occurrence, which normally takes months to years after the transplant. And a chronic rejection class is due to cellular immune response, antibody attack, or maybe a combination of both. Okay, class, this is a very familiar term when pertaining to the immunology of the transplantation practice. We have here graft versus host disease. So, according to Huther McCann's Book of Medicine, the Understanding Pathophysiology, GVHD is not a problem with heart and kidney transplants, but is a problem in bone marrow transplants. The problem occurs whenever there is an antigen difference between the donor and recipient. And in this case, the bone marrow can basically produce lymphocytes which can become sensitized to the recipient's antigen, thus, the graft rejection of the host. And in medical orientation, GVHD is characterized by clinical signs and outcomes such as liver abnormalities, skin rash that looks like measles, diarrhea, wasting, and death. These are the different kinds of graft. When we say autograft, this is the transfer of an individual's own tissue and is regularly accepted by the body. Isograft, or also known as syngeneic graft, is the transfer of tissue between genetically identical individuals. One main example is from identical twins. And in this case class, the graft is usually and medically taken permanently. Excuse me. Heterograft, or also known as xenograft, is the transfer of tissue between different species, and this is always rejected. And lastly, we have homograft or allograft, and this is the transfer of tissues or tissue between two genetically different members of the same species. One perfect example is a graft from human to another human. So, the rejection of the graft by the recipient is called reaction. Sorry, homograph reaction, and its manifestations are vascularization of the graft, mononuclear cell infiltration, marked reduction in circulation, and necrosis. So, homograph reaction class is due to or mainly due to cell-mediated reaction, and the rejection depends primarily on helper T cells, which activate cytotoxic T cells, your macrophages, what else, B cells. But however, antibodies still contribute to the rejection of certain transplants, especially in when it pertains to bone marrow transplants. And that ends our short presentation about the major histocompatibility complex, the human leukocyte antigen. Thank you and have a nice day.